All right, so what leads to antibiotic resistance? Practices. In what way? Can you elaborate a little bit, Molly?
Like, they used to always, like, if you brought your child in for an ear infection and there was, like, some fluid and, like, angry looking ear, they just, like, prescribe antibiotics for it.
Okay, so the default's just prescribe, and being on the parent side of that, I do, there's part of me that appreciated that at the time because Jake had constant ear infections as a kiddo, so. But, yes, absolutely, overprescription. What else?
Prescribing if you don't exactly know what you're treating.
Good. So prescribe. I'm just going to put blindly here as a summary of that, but prescribing without knowing what's actually causing the infection. Good. What else?
Animals.
Use in animals or livestock. Alexi, did you have one?
That's what I was
Okay. We might also include fruits as well. So there are some antibiotics that are used actually to prevent microbial growth or rot on particular food item or fruits and vegetables as well. What else? Taylor.
People not finishing their full antibiotics.
Others? Alexi, go ahead.
I think they talked about like when they're testing the antibiotics, like it gives the bacteria like multiple or like thousands of chances to break the code. Is that what they said?
Yeah, and what would we mean by breaking the code? What is that? How? And what we've talked about in the past couple of class periods, what has to happen to break the code or to become resistant? What's happening to the cell? Mutations. That's the breaking the code. It's an accident, but that allows for that to take place. And those cases in which we give them those multiple chances, well, those come from features like not taking the full dosage. And we'll talk about this idea of the theory in a minute, and what allows for that breaking the code. But there are other examples in which this can take place too. Taking antibiotics after the expiration date. Usually, antibiotics have a shelf life, like most drugs. And after that shelf life, they oftentimes are not as active or as effective as they originally are stated. So for example, if you have 500 milligram tablets of, say, doxycycline, you let it sit in your linen closet for 3 years, and you come back and you measure how much of it is actually still active, it may only be 300 milligrams. And so it reduces the amount of active compound. So taking past expiration. Others?
Sharing your prescription with others.
Yeah, that kind of goes along with this too. Sharing prescriptions. I just get guilty with this lecture because I look at all of these and I'm like, yep, done that, done that, done that. So, any others? Let's take a look at one more. I want to go back to this one real quick. There's actually been suggestions, and Europe has made a push on this, of, okay, so how do we prevent this from happening? So instead of just prescribing to treat whatever is there, what might we propose to do? Let's say you're a clinician or you're a nurse and you are going to go propose to an MD, because MDs don't know shit. Sometimes. Let's say that you want to propose a different way instead of just giving an antibiotic. And oftentimes these prescribing blindly, what type of antibiotic is being prescribed? My apologies to the future MDs in the room too, because I think I have put my foot in my mouth about 5 times already today by making really stupid comments in both classes. So I love MDs, except for when they don't make good decisions. All right. So let's take a look at antibiotic resistance again. One of the things that we talked about is antibiotic resistance, or antibiotics serve as a selective pressure. Okay? And this kind of goes back to the idea of evolutionary theory again. So in this example here, what we're just going to focus on, we're going to focus just on the right-hand side. I don't want to necessarily focus on persister cells right now, because that's not something we talked about. But on the right-hand side, you have a population of cells. Typically, when we are looking at population of bacterial cells, we oftentimes consider them to be genetically identical. But that's actually not the case. There is heterogeneity. And the differences may simply be no more than one nucleotide that's different. Okay? But we've looked at examples where single nucleotide changes, if in the right location, if within a codon of a gene coding region, can have quite a dramatic effect. So we looked at, for example, Neisseria gonorrhea and how single nucleotide changes within the gene that encodes gyrase can lead to increased resistance to ciprofloxacin and other quinolone antibiotics. So what we find is that in a population, if we're treating with particular concentration of antibiotic, and let's take a look at these 2 examples here, not taking the full dose, taking past expiration. These two would be examples where the concentration of the antibiotic within the body would be sub-inhibitory, not enough to inhibit the growth of the whole population of cells. So let's say the MIC for this bacteria is something on the order of 4 micrograms per milliliter, and we're only achieving half of that. Well, there are going to be some within this population that are resistant to that concentration. That concentration may be enough to kill, say, 80% of the cells, but those that are resistant are able to survive. Because they're able to survive, they can replicate, divide, increase in number, and they become the predominant population. Okay? This is that step that leads to antibiotic resistance. And what's different between these cells again, those shown in orange here, the ones that we consider to be resistant, they have a different genetic makeup. In other words, we're selecting with antibiotics for particular mutations that allow for these organisms or microbes to survive in the presence of that antibiotic. Go ahead, Molly.
Okay, so how would that be different if there was a high, because they're already going to be resistant?
Yeah, that's a good question.
Be able to replicate and...
That's a very good question. So when we say resistance, we usually, one of the things that we don't oftentimes do is we don't oftentimes refer to a percent, or like to an amount of antibiotic. You're absolutely right that when we talk about resistant and sensitive, it's binomial. It's either one or the other, but there's not this shade of gray. What we can find is the bacteria might be resistant to up to a certain concentration, but sensitive above that concentration. So let me go back to the Neisseria example real quick. Actually, I'm going to try to pull up these slides. Let's see if I can find this real quick. Okay. So if you'll remember from the example that we looked at, this was one of the last slides in the example of Neisseria gonorrhea. Oh, now this doesn't want to increase in size. And this represents the gyrase gene and just a couple of different portions of it. So one is gyrase A and one is PAR-C, which is another gene that actually has gyrase-like activity. And look at what happens with the increasing number of mutations. And we kind of pointed this out towards the end. So this one has a single mutation in gyrase. All of these have 2 mutations in gyrase. And then the last ones have additional mutations in this PAR-C gene, which is also targeted by ciprofloxacin. So as the number of mutations increases, Molly, what's happening to the MYC? Yeah, go ahead. What happens to the MYC?
It increases.
It increases as well. So here, if we were, for example, using, let's say, 4 micrograms per mil of ciprofloxacin, it's going to inhibit the growth of all of these strains that have 3 or fewer mutations, okay? But it would select for the growth of those that have 4 mutations. So let's talk about how this actually would have progressed for a minute. Likely, what would have happened is you use a concentration of antibiotic, say, in this, let's say we're doing a broth dilution experiment and we use 0.015 micrograms per mil of the antibiotic. The bacteria undergo replication many, many times. Oops, one of them undergoes replication and gains a missense mutation in gyrase. Those will now grow in that 0.015 microgram per mil. Everything else is going to die off, though. All right? Now we need to treat with a higher concentration of antibiotic to inhibit the growth of these mutants. And if we do that, replication would still continue for those that have gained a second mutation. And so what I'm trying to argue here is that we would continue to increase or to continue to push ahead the concentration of antibiotic that we would use. And the organisms that are able to divide in that concentration would be ones that are gaining additional mutations. Okay? It's very unlikely that we would find, if we were treating Neisseria gonorrhea with 0.015 micrograms per mil of an antibiotic, it is very unlikely we would find a strain to arise that gains 4 mutations all at one time. Because there's no selective advantage for having all 4 mutations at the same time. That only becomes advantageous as the amount of antibiotic increases. Does that help a bit? This is a very challenging. This can be a very challenging concept. So I just want to.
Almost like, let's say, maybe this is an oversimplification, but like they make an antibiotic that is like made, like they've taken into account like that, and so this at this dosage we know that it will kill that. So if you take it at a lower dosage, you're just sort of like almost going back to like only killing the lower.
That's exactly right. That's exactly right. So oftentimes what we are trying to achieve is to treat with a concentration of the antibiotic that's much higher than the MYC. Okay, with a concentration that would not only inhibit those that show some intermediate resistance to the just treat with a high enough concentration that it's going to kill everything. That's the design. But if we start treating with lower concentrations, by not finishing a full dose, by using expired antibiotics, then that allows for those that are resistant to that concentration to become the predominant organism. Okay? Let's take a look at this in a different way, and maybe this will help too. So here is another example. Here's a population of cells, and again, if we were to look at any one single bacterial cell, like usually in a culture of E. coli, we've got 10 to the 9th cells per milliliter. If we were to look at any one single cell, its minimal inhibitory concentration would be different than the MYC of another single cell. And that's what I'm trying to show up here. there's population heterogeneity. In a population of bacterial cells, you have some that are particularly sensitive to an antibiotic, some that are particularly resistant to an antibiotic relative to the rest of them. When we perform minimal inhibitory concentration experiments, oftentimes we are looking for what's the minimal inhibitory concentration that it takes to kill all of these. So our concentration that we're typically using is way up here. If we treat with a sub-inhibitory concentration, like this here, who's going to die? Or who's not going to grow? So let's say and I'll put numbers to this too. Let's say that this is 1, 2, 4, 8, 16, 32, and 64 micrograms per mil of an antibiotic. So let's say we're treating with 32 micrograms per milliliter. Which of those are going to be inhibited in their growth? Those to the left or those to the right? Those to the left. Those that are all more sensitive, where it takes even less antibiotic to inhibit their growth. So what we're saying is, if we treat with 32 micrograms per mil of antibiotic, all of these cells shown here, shaded in brown, they're going to either die or they will stop growing. Those that are resistant to, or those that are resistant to concentrations at 64 to 32, they now are able to do what? At 32 micrograms per mil of antibiotic, what can they do? They can grow. They become the predominant population. And they are the ones then that pass along their genetic information to the next population, or to their daughter cells. So what we find in this case then is a shifting population. Like this. So those that pass along their genetic information to the next generation might have a phenotype that allows for them to survive those higher concentrations of antibiotic. Since they're able to, and that, remember that phenotypes, protein expression is always associated with what are the genes, what's happening in the genotype. So overall, we find a shift in the population, and now as a whole, is this population more resistant or more sensitive to the antibiotic relative to the original one shown in black? They're more resistant. So that's how this happens. Okay? So the key thing to keep in mind here, this is a bit of a change from how we've been thinking about things, or how we even observe results. We've thought of it as E. coli is E. coli is E. coli. The strain we use is genetically identical all the time. That's actually not the case. There's variation in there, and the variations are what's allowing for events like this to take place. I'm going to pause for a minute, and then and give you guys a minute to write, or do whatever you need to. Take a drink. Are there questions on this concept? Okay. Let's see if there's anything else I want to cover here. Keep that in mind. We're going to come back to it towards the end. But I just wanted to point out real quick then, here is what the CDC considers. This slide is a couple of years old now, but these are the organisms that the CDC, Center for Disease Control, consider to be the biggest threats right now with respect to antibiotic resistance. So it breaks them down into hazard level urgent, hazard level serious, or hazard level concerning. And these are placed into different groups based on factors like, well, do we still have other antibiotics that we can use to treat these organisms? how virulent or what are the symptoms of the illness? Are they ones that cause mortality, or are they ones that are more of a nuisance? How fast does the disease spread? Those are the factors that they're considering here. And if you take a look at these lists, top one on there, what's number one? C. diff. Anybody want to take a guess as to why? It's typically hospital acquired. It's typically found in immunocompromised individuals. It's oftentimes the result of treating with broad spectrum antibiotics. And we don't have a lot of good antibiotic treatment. And the success of antibiotic treatment against it is actually really low. Only like 30% successful in removing the infection, aka poop transplants. Carbapenem-resistant Enterobacteriaceae. Carbapenem is a beta-lactam. Drug-resistant Neisseria gonorrhea, this cephalosporin resistance. So what you'll see in this list is that the organisms that are listed here are all ones that are associated with some sort of resistance marker or resistance to an antibiotic. Multi-drug resistant Acinetobacter, drug-resistant Campylobacter, fluconazole resistant Candida. This is a yeast infection. Okay, so if you know somebody who has had thrush, for example, caused by Candida. Extended spectrum beta-lactamase producing Enterobacteriaceae, E. coli, Salmonella, Klebsiella, that all produce beta-lactamases that can inactivate beta-lactam antibiotics. These have all arisen from events that we've talked about. Some are these, and it's kind of a case of bad luck. It's not just these alone, but it's these plus genetic events, mutations in the genome passed down from one generation to the next through vertical gene transfer, or exchanging of genes, gaining of genes through one of the horizontal gene transfer mechanisms, conjugation, transformation, or transduction. So we could put together a list very easily of what do we consider then to be the things that we need to do to prevent these infections from spreading, or to at least slow the rate of antibiotic resistance. This is not necessarily a complete list by any means, but these are right now considered to be some of the best policies. So as a patient, self-advocate, speak up for yourself. If a doctor is proposing for you to go on a broad spectrum antibiotic, question why. Okay. Wash hands as a way to stop spread of bacterial spread. This one should be pretty much self-explanatory. No signs and symptoms of infection. In other words, what are those called? Antibiotics. Do they work against viral infections? No, they have no effect whatsoever. So if you are going in to the doctor for flu, don't ask for antibiotics. A good MD will not, or PA, nurse practitioner, any of those sorts, will not give you an antibiotic for that, because it will not help. It'll probably make things worse. As healthcare providers, be knowledgeable, be aware, know what types of infections are out there, and know some of the themes that we've talked about, or some of the theories. Broad versus narrow spectrum. Okay? Consider informing patients, even though as patients sometimes we get tired of hearing things like make sure you take all of those antibiotics, or make sure you throw away any of the extra ones afterwards so that they're not sitting around at the house. Those are things that honestly patients need to continue hearing. And then in order to protect patients, and this becomes an important one when we talk about like C. diff infections in particular, wearing things like personal protective equipment, throwaway gowns or throwaway gloves that once you are exiting a room, you can just throw them into a hamper and have them incinerated instead of washing them. Okay? Washing hands before and entering and leaving instead of just using hand scrub, but actually physically washing seems to be just as effective. And then removing temporary medical devices when not needed. For example, catheters. Catheters are very prone to bacterial infections. because bacteria can form biofilms on them and they become really challenging to treat at that time. So there's been more common use now, or what we're seeing more commonly is use of disposable catheters rather than washing and reusing them. Emma.
I feel like this is kind of like a devil's advocate question, but like when I did my clinicals, the only room we had to physically wash our hands for was C. diff. But like being on a med surg floor, even if you're not in a room with C. diff, you have people with weakened immune systems who are already on a ton of antibiotics, who have open wounds. Like why? Like there's a big push now, like I think in hospitals and stuff, that you can use hand sanitizer on the way in and on the way out, and that like that's enough.
Yeah, it's a good question.
Like if you're looking at this, though, it's like, is it really?
Maybe this is just my personal thought, that hand washing is more effective. And the reason why I say that is because of the reason like C. diff. If that's what they're doing in hospitals, then what I would be always asking for in a case like that, and this just comes from my way of thinking about things, I'd be asking, well, why is that best practice? What have you done to figure out that that is just as good as saying we're going to wash hands coming in and out of every room? That would be where my thought process is. My guess is that is the process because C. diff, we know 70% ethanol does not kill spores. And that's what we really need to be concerned with C. diff. As soon as it hits oxygenated environments, it sporulates. And those spores are really challenging to kill. But once it goes back into a human being through ingestion or through, God forbid, a wound, it's usually through ingestion more than anything else. What we're going to find is that it releases its spore coat, it begins to go into a vegetative state and multiply again. So I think that what I would be doing is just asking why are we sure that this is best practice, or what's the evidence there to support that this is best practice, right? Many of those other types of infections that we would find typically associated with hospital or individuals who have come out of surgery are going to be skin infections. Staph, Pseudomonas are going to be the primary ones, and those are organisms that don't necessarily undergo they don't undergo a sporulation. So you would assume at least that ethanol should work. At least, I mean, that's the general assumption, right? So that's my guess as to why that is. But I would be of the mind of, no, just wash all the time. That's my personal preference. But thanks for bringing that up. And I think those are the types of issues that, for the majority that are in this room, the majority of folks in this room are individuals who, whether you're doing nursing or something else, you guys are thinking of health-related issues. Those are the questions that we need to be asking. Yeah. Any other questions? Okay. Let's try, well, actually, real quick, before we go any further, does anybody have any questions on things that were brought up in the TED Talk, or any comments on the TED Talk itself that you wanted to address? Okay. I really appreciated the comments. I thought that the wikis were really, really good, and your guys' comments were excellent for that one. I always enjoy watching that video. I'm going to open up a review question then. Okay. What I'd like you to do is, focus here primarily on the first and the third of these bolted questions, because these are examples of what you might see on this coming up exam. Okay? So the first one is, explain how a population of Staph aureus may gain resistance to a low dose of methicillin. There's actually a couple of potential ways, but I'd like you to just come up with one way and use today's conversation as kind of the context for that. Okay? And then the second one up there is more application based in terms of everyday life, I suppose. So patient with streptococcal sore throat, in other words, they've got strep throat. Take penicillin for 2 days of a prescribed 10-day regimen. Because guys are bigger idiots and would be the ones not to take the full dose of the antibiotic, I suppose because he feels better, he then saves the remaining penicillin for some other time. After 3 more days, he suffers a relapse, a sore throat. Discuss the probable cause. Okay? So begin with either of those, but I'd like you to take the time to at least jot down some ideas for responses on both of them, and then we'll take maybe, we'll take about 4 minutes. Let's go with this for about 5 minutes, and then we'll talk for a little bit to just see where we're at with this. And feel free to talk it over with somebody else if you need help on this too.